Good evening, everyone. Another beautiful Tucson evening in March. And welcome to the Science Cafe, brought to you by the U of A College of Science and the Flandreau Science Center. Please give a big round of applause to Luke Pangle.
All right, so this is a really fun venue to give a talk. So my name's Luke Pangle. I have a master's degree in tree physiology, and then I did a PhD in hydrology at Oregon State University. And then just in September of this year, I had the pleasure of moving here and beginning to work on what I think is undoubtedly the most fascinating laboratory experiment in the earth sciences on the Earth. And I'm happy to tell you about that tonight. Um, right away, I want to acknowledge that I'm very much a beneficiary. Having come here in September to begin work on this project, I want to acknowledge that before me, there were many people whose generosity and also their creativity led to the development of this project that I'll tell you about. Foremost is Mr. Ed Bass, who was the former owner of the Biosphere 2 before donating it to the University of Arizona. And he has also provided a charitable financial gift that has made possible the Landscape Evolution Observatory to date. And then also listed here is just a long list of names. You can't read through them, but just to show you that a whole wide group of scientists at the University of Arizona and also at other institutions in the US and internationally all contributed either in thought or in action to the development of LEO. So I want to acknowledge them. And also Paul Ingram and Shippard, who did a lot of the photo and video documentation of the construction of the Landscape Evolution Observatory. Many of the photos I'll show in this presentation were taken by Paul, and there's a lot of great video footage on our website as well that you can check out afterwards if you're interested. All right, so I'm going to tell you all the dirty details about the Landscape Evolution Observatory, or LEO, which is an experiment that includes 3 artificial landscapes that have been built inside the climate-controlled facility of Biosphere 2, where we are conducting research to better understand how landscapes change through time and how that landscape change through time influences the way those landscapes function, the way they cycle water, carbon, and energy. But before I go into all the details, I want to give a little bit of context about, what was the motivation for LEO? What are the scientific questions that we believe can be answered there in a more effective way than they ever could anywhere else? So I'll first do a quick primer on the hydrologic cycle in southern Arizona watersheds to bring everyone up to the same level. And then I'll go into those motivations that were the underpinning for developing the Landscape Evolution Observatory. And then I'll tell you all the details about how it was built, what we measure, what LEO does, and then I'll close by showing you some of the early results from some initial experiments that have been done there thus far. All right, so first, these are 2 Google Earth images. The one on the left is, I don't think you can see it, but it's of the Marshall Gulch, which is a big watershed up near Mount Lemmon. And this is an area where University of Arizona scientists have been conducting research for several years now. And the one on the right here is actually the Agua Caliente Canyon and hillslope area just on the northeast side of Tucson. And unfortunately, it's not extremely clear, but I put these 2 contrasting images up here as examples of how landscapes vary in space across our extremely heterogeneous environment here in southern Arizona. So first, they have quite different climates. Of course, as you go up in elevation to Mount Lemmon, catchments near the 8,000 feet elevation mark have much more precipitation than at lower elevations, and they also have cooler temperatures. So I think everyone's pretty well aware of that. One distinguishing feature of these landscapes, when you compare these high elevation environments to our lower elevation environments, is that there is a longer distance to the valley bottom. So what's that? Well, again, I don't think you can see it so clearly, but You're probably familiar with what the landscape looks like here in our lowland hillsides. It's very carved up, you might say. If a raindrop falls anywhere on this landscape, it doesn't have very far to travel before it gets down in a gully, places where you have the formation of channel flow and very rapid movement of water off the landscape. In contrast, these very high elevation watersheds like Marshall Gulch up near Mount Lemmon, they have a much longer distance to the valley bottom. So a raindrop that falls anywhere on that landscape has a much longer distance to travel before it ends up in a stream channel and gets moved out of that landscape. And that has some significance that I'll elaborate on a little bit more in some later slides. Another really important feature is that in these higher elevation watersheds, you have much deeper soil than you do in the lower elevation watersheds. About 3 feet deep at the Marshall Gulch at Mount Lemmon, whereas at the Agua Caliente hill slopes and canyons, the soil might be only 1 to 2 feet deep. And last, and perhaps most obvious, is there's a vast difference in the plant biomass. At those higher elevations, we of course get into conifer forest biomes compared to the scrub or desert vegetation of these lower elevations. All right? And along with these different features of the landscape that we can observe changing in space, there's also a lot of variability in the way these landscapes function. Now, when I talk about the way a landscape functions, I'm essentially talking about three things. The way that landscape cycles water, carbon, and energy. So the water cycle, of course, is referring to the way in which all the precipitation that falls on a landscape gets partitioned between evaporation back into the atmosphere, storage of water in the subsurface, or groundwater recharge or stream flow generation. Similarly, there's a carbon cycle that begins with the assimilation by vegetation of carbon dioxide gas out of the atmosphere, which they assimilate into their leaves to make carbohydrates, plant food. Additionally, there's respiration by plants and also microfauna. So when they break down sugars, they exhale essentially just like we do, emitting CO2 into the air. Carbon can be stored for long periods of time in biomass like wood and also in the soil. And then additionally, inorganic and organic forms of carbon get exported out of watersheds through the stream flow. And last, landscapes also cycle energy. So we have all this incoming energy from the sun. Some of that radiation gets reflected from the land surface back to the atmosphere. Some of it goes to heating the Earth's surface, and then the Earth's surface then emits thermal radiation back into the air. And very much of that energy goes towards a phase change of water, evaporating water from the liquid to the gas phase. So now let's look at some ways in which the function of these different types of landscapes vary, looking just at the water cycle. So this is a Google Earth image of a catchment near Tombstone, Arizona. It's referred to as the Walnut Gulch catchment. It's a place where the United States Department of Agriculture has been doing research for a couple of decades now. And it's a lower desert habitat. At the bottom of the catchment here, there's a large weir, a large structure that can be used to measure the total volume of water flow out of the catchment and also sediment. And so in this particular watershed, they get about 12 inches of rain per year. All right, and that's dispersed over storms throughout the season, mostly, as you know, in the monsoon season in July and August. Of that 12 inches, about 10 inches infiltrates into the soil. And it doesn't stay there long because of the high energy at the Earth's surface here in the desert southwest. Essentially, all that water gets turned around pretty quickly. It's evaporating back into the atmosphere, either evaporating from the bare soil or evaporating from plant surfaces after being taken up by the plant roots. So that's 10 inches out of 12 inches on an annual basis. It's basically infiltrating and going right back into the atmosphere. The other 2 inches falls on the surface, and a significant amount of that flows directly over the land surface in sheet flow or through small rivulets on hill slopes and eventually into gullies or washes, like shown here. And of that amount of water that goes into the gully, much of that infiltrates there in the gully. And of that infiltrates, most of it's going to get turned back around as well. Riparian vegetation is going to take that water up, transmit it back to the atmosphere. And in this particular catchment, the Walnut Gulch catchment, about 1/4 of an inch per year actually flows out of their catchment. So not too much. Now let's contrast that with this dark image, which again is the Marshall Gulch catchment. Here, there are major differences, beginning with precipitation. There's about 32 inches of precipitation per year at this site, almost 3 times more than at that lower elevation Walnut Gulch catchment.
I'm thinking we can go back and look at some of these images later once it's a little darker.
Sure.
But if we were able to see this image, can you just describe what we would see? Is this like forest that we're looking
Definitely,
at? Or?
yeah. So what we have is one big catchment, basically consisting of 2 large hill slopes, and then a stream channel that drains Marshall Gulch, which is kind of running right down the middle here. And this is up near Mount Lemmon, so it's covered mostly in ponderosa pine forest. And the stream that drains Marshall Gulch joins Lemon Creek and eventually flows down the mountain and out of Sabino Canyon. So this is one of the major tributaries to the Sabino Canyon. And so at this higher elevation site, you got more precipitation. And here is a first drastic difference. All that precipitation infiltrates into the soil. Now at that lower elevation site, you saw that only 10 inches infiltrates and another 2 inches rapidly runs off the surface. Here, all that water goes into the soil. And not all of it immediately evaporates back into the atmosphere. Some does, but on an annual basis, about 19 inches, not the whole 32. So that's a drastic difference. That means that an additional 13 inches infiltrates deeper into the soil, beyond the root zone of the plants, recharges shallow aquifers, and eventually contributes to stream flow out of that catchment. And that stream flow in these high elevation catchments is really important for us relatively low elevation dwellers, because the water that's generated in these high elevation catchments is actually the primary source of water that recharges the aquifers here in the basin through 2 separate processes, one called mountain block recharge and another called mountain front recharge. So with this kind of satellite imagery, with geographic information systems, and also with a variety of different types of field measurements, we can go out and measure differences in these landscape characteristics. And we can also see that those differences in landscape characteristics are associated with changes in the way these landscapes function, the way they cycle water, carbon, and energy. But as earth scientists, we want to go deeper than that. We want to answer the question, why? Why are these landscapes functioning so differently? And what processes led to their different functioning? Okay? But getting at those questions of how and why is really challenging. And to communicate to you why that's so challenging, I wanted to use an analogy. So I thought really carefully about this, and I decided that the best analogy is beer. So I love beer. Apparently a lot of you do as well. My wife and I just began trying to brew our own beer at home, unsuccessfully. We ended up with something more like Robitussin. But just consider for a second that you tonight you sample some really excellent beer, and you think, I want to recreate this. I want to make my own beer, and I want it to be like this. Well, immediately you're confronted with a lot of questions. You know, what kind of malt do I use? How much malt do I need to mix with a particular volume of water in my wort? How long am I going to boil my wort? You know, what kind of hops am I going to use? How much? And when do I add them in the brewing process? How much yeast am I going to have to add to get this thing to ferment? And how long is it going to ferment? So you've got a lot of questions that answer right off the bat. And what you have is simply, for guidance, is a final product. And what you really need is a recipe. You need to know how all those parts go together. And so as earth scientists, we're confronted with exactly the same dilemma. When we want to explain how it is that landscapes came to be so different and how it is that they came to function so differently, we're essentially looking at a finished product, or at least one discrete version of a continuously evolving product. We're looking at a landscape that has shaped through time as influenced by the parent geology, the different rates of factors that influence soil formation. We're looking at differences in climate and also differences in vegetation. So how all those pieces come together to influence the ultimate form of a landscape and the way it functions is a really challenging thing to figure out. So when scientists at the University of Arizona were presented with the opportunity to create a new experiment at Biosphere 2, something that couldn't be done anywhere else and that would utilize first the enormous space in the facility and also the climate control capabilities within the facility. When presented with that opportunity, they asked the question, what if rather than observing the finished product, that is going out and measuring landscapes as they exist today, what if instead we could build a physical model of some portion of a landscape inside this climate controlled facility and build that physical model of this portion of this landscape in a relatively immature stage of development? Then we can observe through time how that landscape evolves as we are controlling the climate, controlling the amount of rainfall that goes on this landscape, and ultimately controlling the type of vegetation that gets introduced into this landscape as well. So from that motivation and that question was born the Landscape Evolution Observatory, or LEO. So LEO is the world's largest earth science experiment aimed at better understanding how water, carbon, and energy cycle through landscapes. Inside the Biosphere 2 were constructed 3 large landscapes, which we refer to as zero-order basins. This is essentially an element of the landscape that has topography, it has convergence into like a gully area, but it doesn't support a perennially flowing stream. And then more commonly, we just refer to them as hill slopes. So each of these hill slopes, like you see in this image, is 30 meters long in the upslope dimension. That's about a little over 100 feet. And 11 meters wide in the cross slope dimension. That's about 33 feet. And in total, there is a 1 meter depth of soil installed within each of these hill slopes. So we've got 330 cubic meters, or about 500 metric tons of soil in each of these slopes. Each of the slopes is built so that the overall angle is a 10-degree slope, but down the center of the slope you have this gully area, so in that part of the slope there's steeper parts of the hill slope. All right. Each one of the hill slopes is contained inside an enormous steel structure. This is looking at one of them from the floor space inside Biosphere 2. And these were constructed by M3 Engineering, who came in and helped build the site under the direction of the researchers who were working at Biosphere 2 at that time. And so all 3 of those steel structures, all the soil that they contain, the actual hill slope, is then contained inside this 10,000 square foot plus portion of the Biosphere 2 facility, which is located near Oracle, Arizona, just in your backyard here. Inside each of these 3 large bays, which you can see inside this wide-angle photograph here, is one of the hill slopes. And there's also an entire air circulation system. So the air can be circulated through a basement and then up through a ventilation system and down over the hill slope. And the temperature and the relative humidity and the wind speed of that airstream can be controlled individually for each slope. So we have that kind of climate control. Also on each slope is a custom engineered irrigation system. On vertical risers attached to the side of each of those steel frame structures, we have an array of different sprinkler heads that are connected to independent plumbing circuits. And with the whole array of different sprinklers we have, we can rain on these hill slopes at rates as low as about 3 millimeters per hour, so a nice little drizzle, up to about 45 millimeters per hour, or almost 2 inches per hour, simulating what's more close to a monsoonal thunderstorm that we experience here in the summer. So we can control the entire climate. Now, one of the motivations for this was to try and build a landscape that at time zero was, as closely as we could make it, uniform across its entire area and volume. So to do that, the scientists had to decide what kind of soil material are we going to use. And so ultimately they decided to use this basaltic tephra, which you see here. So this is a volcanic rock that's emitted from a volcano, and it was extracted from a mining site near Flagstaff, Arizona. The scientists worked with a mining company to then take this enormous mass of this basaltic tephra and grind it down to a texture that more closely resembles a loamy sand type soil. That loamy sand type soil, all 1,500 metric tons of it, was then transferred to Biosphere 2, moved inside the climate controlled facility, and many different workers began to fill these enormous hill slopes with this soil material. And they worked day and night, as you can see here, working through the night. And as they filled these steel frame trays with this soil, you can also see all these vertical posts here. Each of those marks one location where there's a vertical transect, or a vertical profile, I should say, of different sensor types that allow us to monitor different environmental variables in the subsurface. Here's an example of a worker installing one set of sensors in the soil. So what kind of sensors and how many? So here's a diagram, and I'll run through a couple iterations of this diagram. There are 5 different panels here. Each one of these panels is meant to represent a different depth within our soil. So you can see at the top, 5 centimeters, 20, 35, 50, and 85. And I realize I'm in all metric here, and most of you probably think in inches, but that's about 2 inches, about 8 inches, about 15 inches, all the way down to about almost 30 inches at 85 centimeters. So at 5 different depths in the soil, and the dimensions along the 2 axes here show the actual dimensions of our hill slopes, 30 meters by 11 meters. And so everywhere you see a black dot here is a location at each of those 5 depths where we have sensors that measure water content, so the actual amount of water stored in the pore space of the soil in that small area. We have other sensors that measure the potential energy status of that water. So if you can measure the potential energy status of water at multiple points in space, you can determine where gradients exist to drive the flow of that water, and it'll tell you the direction and also the potential velocity at which the water might flow. So that gives us information about where the water is going to flow and how fast. At this same 496 locations where we saw the black dots, we have these devices here which are called tension lysimeters. These are basically ceramic cylinders that are hollow on the inside. Now they're made of ceramic, and that's porous. So water in the soil will actually absorb into that ceramic. Each one of these devices is attached by a thin plastic tube to a syringe that's actually located outside the hill slope. So we can take that syringe, pull on it, and it creates a slight vacuum inside the syringe. And that slight negative pressure causes water in the soil to flow through the pores in the ceramic into the hollow area inside, through the plastic tube and to our syringe. And that way we can actually pull samples of the water that exists in the soil pore space, take it into the lab, and measure its chemistry. So that's at 496 locations. In each of 3 slopes, we have these types of sensors. So here now I've added these red diamonds you see here. Those are just at the soil surface, and those diamonds represent the locations of what we call heat flux plates. These are instruments that allow us to measure the actual rate at which heat is being transferred from the atmosphere into the soil. So we can carefully monitor that exchange of energy between the atmosphere and the land surface using these types of instruments. Now I've added these purple boxes that you can see at 4 of the 5 different depths. There are 141 of those in total. Those locations correspond to soil gas samplers, and these are these thin white tubes that you see here. That's basically a tube that's made of a plastic material that's porous to gas but not to liquid. And so in the same way we have those liquid water samplers, we also have these gas samplers that we can extract a sample of the gas phase in the soil pore space and also measure its chemistry. For example, measure the amount of carbon dioxide that's in that total amount of air. Okay, and now I've added these green circles, which you see here and here at 4 of the depths, and also these blue triangles. The green circles correspond to these soil gas sensors that measure the CO2 concentration of the soil gas phase. Now, what I showed you before are devices that we can go and manually sample, pull one sample just at that instant. These instruments are hooked up to a data logger, and at every moment in time, they're monitoring at 48 different locations in the subsurface what is the CO2 concentration of the gas. By having those measurements, we can quantify where the gradients exist in the soil that would drive the flow of that CO2 gas, either back into the atmosphere or from the atmosphere down into the deeper soil. And last, those blue diamonds that came up here on the right side, these are pressure transducers. They're installed at the base of the soil profile in the steel frame tray. These devices, when submerged in water, will tell you the exact height of that water in which they're submerged. They're the same type of device that's used in stream gauging, so to monitor water levels in rivers. So when we have a water table that forms, that is a zone of complete saturation of the soil pore space, we know spatially where that water table exists and how deep it is. So all these instruments in total add up to about 1,700 different sensors or sampler types that are buried within each of these hill slopes. So an amazing ability to monitor everything that's going into the subsurface. A better job than we could ever do if we went out into nature, where the system is not contained as it is in Biosphere 2. Now, perhaps the most fascinating aspect of this entire structure is that each of these steel frame trays, all the soil within them, all the sensors, and any water we add through our irrigation system is weighed at every moment in time. We had Honeywell Corporation design custom load cells. These are essentially very high capacity scales to measure mass. And each one of the slopes rests on 10 of these. They're shown here covered in insulation, and that's to reduce their exposure to daily fluctuations in temperature. Those allow us to monitor the total mass of the slope at every point in time. So here, for example, on this graph, on the x-axis is time, going from February 25th of last year all the way to April 29th. So in April, we did a series of experiments where we rained on the hill slopes, and you can see those load cells responding. As the rainfall infiltrates in the soil, the total mass increases, we see that. And then as that water begins to drain out of the hill slope, the mass decreases. Now, the really amazing capability this gives us is it allows us to know the total amount of water that's stored in the subsurface at all times. So if you remember, I pointed out that the water balance consists of evaporation, storage of water in the subsurface, and runoff. Now, evaporation and runoff, we have means to measure pretty well in the field. But at a field site, like in the Catalina Mountains, you could never know how much water was actually stored in the subsurface and how that's changing through time. These load cells give us the capability to actually monitor that. And you can see here the weight in pounds. On average, we're a little over 1 million pounds, considering the structure, all the soil and water, and all the sensors installed inside them. So that's a really fascinating capability we have. And I mentioned that all the water that flows out of the hill slope, this is analogous to what would be stream flow in a natural environment. We monitor that as well with a series of gauges that measure the outflow from the slope. So just as you can monitor the volume of water flowing in a stream, we do that for our hill slopes as well. All this instrumentation really allows us to see inside these hill slopes. Many of these processes that are going on in the subsurface, the flow of water, the movement of carbon dioxide gas, ultimately what will be uptake of water by roots, we can use this dense array of sensors and the data that's derived from them to produce 3D imagery like this to show us exactly what things look like in the subsurface. This is one snapshot in time of an experiment that was done in February of last year, where we rained on the slopes, the slope became saturated. You can see by the blue color here that the slope became more saturated in that gully area. And after some period of time, we actually had the formation of overland flow and some gully erosion. And we're studying how that gully erosion process developed through time. But having this array of sensors allows us to visualize the subsurface in a way we could never do in nature. And not only the subsurface. At present, we are currently working to install instruments above these hill slopes as well. These are instruments that will allow us to monitor the microclimate above the slopes, that is the temperature, the relative humidity, the wind speed, and also the amount of light coming through the biosphere glass and onto the hill slope surfaces. Here you can see Michael Sabayan, our lead instrument engineer, and Nate Abramson, our research technician, using rappelling gear and a gantry crane system that's built onto the hill slopes to access the slope and install this instrumentation without actually walking on the hill slope. And this way we don't have to disturb the surface through time. The instruments we're installing look much like ones you've probably seen before. We have an anemometer, which measures wind speed and direction. We have temperature and relative humidity sensors, and as I mentioned, light sensors. So as you can see from these photos, at present these hill slopes just have a bare soil surface. So for the first 2 to 3 years of this experiment, we are really going to concentrate our research on understanding the hydrology of these hill slopes just in a bare soil condition. But eventually, we are actually going to introduce vegetation onto these slopes. We're going to disperse seed on the slopes, as would occur naturally in an environment in the Catalina Mountains, so that germination can happen, natural competition processes between the different species happen, and we have the evolution of a plant community. And we'll be able to observe how that plant community evolves and how the evolution of that plant community influences the way water, carbon, and energy are cycled through these hill slopes. So, I'll wrap up here with some initial results from one of the first experiments we've done on the slope. One of the motivating questions for this experiment is how old is soil water, and how long does it actually take water when it enters the soil as a raindrop to eventually end up in an aquifer or a stream channel? And why would we care about that? Well, let me give you another analogy to try and explain what our rationale is for wanting to know that. So Wednesday morning, you get up, and after a night of drinking beer down at Magpies, you're feeling not as sharp as you might otherwise. So you decide maybe a cup of tea will help you get going. So you boil your water, and you take your tea bag, and you drop it in the water. And immediately you start to see things happening. A lot of the compounds that were in those dehydrated tea leaves immediately begin to dissolve into that water. Now, if you pull the tea bag out after 10 seconds, you're going to have a pretty dilute cup of tea there. If you leave it in longer, of course, more and more of that exchange is going to happen of those compounds from those tea leaves into your water, and you'll get a richer cup of tea. And eventually, if you left it in there much longer, it wouldn't make any difference. At some point, everything's going to dissolve into that solution. So the flavor of your tea is very much a function of the contact time between that water and the tea bag. Now, in catchments, a very similar thing is happening, and it's important. So when raindrops form in the atmosphere, that's essentially a distillation process. Those raindrops are just about pure water, although they do tend to form around particulate matter in the atmosphere, so they're not completely pure. That pure water enters the soil, and the longer that water is in contact with the soil, the more the chemistry of that water will change. So the residence time of those water droplets as they enter through infiltration and eventually make their way to the stream channel is very important, because the longer the water's in the subsurface, the more likely it is that a plant can take up that water, supporting the ecosystem function and supporting the plant growth. And also, the longer that water's in contact with the soil is going to influence the chemistry of that water and the chemistry of the water that eventually ends up in our stream channels. So we wanted to investigate what are the range of transit times of water molecules flowing through these hill slopes. So to do that, we take advantage of a couple of key capabilities at LEO. So we have this irrigation system where we can control the actual amount of water that goes on these hill slopes at any point in time. We also have the great advantage that we can manipulate the chemistry of that water. We can introduce what we refer to as tracers to study flow paths through the subsurface and the time scales associated with those different flow paths. So in this experiment, we used a stable isotope tracer. So water, of course, is made up of 2 hydrogen atoms and 1 oxygen atom. But not all water molecules are exactly the same. Some of the hydrogen atoms that contribute to water molecules have a slightly different mass, and we refer to those as isotopes. And it's enough to say that we have instruments that can allow us to measure the different isotope composition of water samples. So at B2, we have a well, and we pump water from that well through a reverse osmosis system to purify our water that goes on the hill slopes. All that irrigation water has an isotope composition with a value of around -60. Don't worry about the units or what that number means. Just know that it's about -60. And that's shown here with these purple dots. This is where we were collecting samples of that water from our irrigation system, take it into the lab, measure its isotope composition. So the first day, we rained on the hill slope for about 6 hours, and we put about 3 inches of rain on this hill slope. And we did that using our typical water with isotope value of -60. Now the very next day, as indicated in this graph here at the top, we applied another rainfall pulse, about another 3 inches over another 6 hours. Now in that case, we went downstairs to our big storage tanks and we added some isotopically enriched water to that storage tank. So the water that we applied in that rain pulse on that day actually had an isotope composition near 0. So different by our typical water by 60, okay? So -60 the first day, 0 the next day. Then, as that water infiltrates into the soil and as it eventually flows out of the hill slope, we're using our sampling devices to pull water samples from the soil pore space, and we're pulling water samples from the hill slope outflow. We take those to the lab and we measure their isotope composition. So at every point in time, those water samples tell us at that point in space and at that point in time how much of the water is coming from this rain pulse, which has a unique isotope composition, and how much of that water is coming from this rain pulse that we applied on the next day that has a totally different isotope composition. So what did we see? We applied those 2 pulses. Here in this middle graph shows the hill slope outflow throughout 2 weeks in April. So after that second rain pulse on the next day, we started having outflow from the hill slope. This would be like increasing stream flow in a catchment. And that outflow persisted. About 10 days later, we applied another 3 inches of rainfall. That again was with our typical -60 water. And in this graph here, these blue dots represent water samples that we were taking from this hill slope outflow and measuring its isotope composition. And what you can see is that over 2 weeks, 2 full weeks, this hill slope is just draining and draining water. The water that's draining out of the hill slope has an isotope composition that looks entirely like our typical irrigation water. That is, that 3-inch pulse of rain that we put in there on April 14th, none of that water is coming out of the hill slope over those entire 2 weeks. So we can also look inside the soil. So with our tension lysimeter devices, we can pull water samples from the soil. This graph shows those samples across the same period of time at about what is 8 inches in the soil and then down to about 30 inches in the soil. And you can see that after we applied that pulse on the second day, at 8 inches in the soil, the water goes from looking like -60, what it would typically look like, to looking more like that isotopically enriched water. In contrast, at 30 inches deep in the soil, that doesn't happen. The water samples continue to look like our typical water. And it's only here 10 days later that we begin to have some samples showing up that are beginning to divert towards that value of 0, that isotopically enriched pulse of water we put in there. So what this experiment shows us is that even after applying 6 inches of rainfall to these slopes, a significant portion of the total amount that would happen in a year in the Catalina Mountains, most of the water associated with that rain pulse on April 14th is retained within the hill slope. So the residence time of water in these hill slopes is on the order of weeks to months. That is drastically different than the scenario I described for you earlier when I was describing the Walnut Gulch catchment. There, water falls on the landscape, it quickly evaporates to the atmosphere. The fraction that does eventually go to a stream channel also evaporates back to the atmosphere. That happens on the order of hours to a few days. Here in these hill slopes, which are more like those catchments up in the high Catalinas, water's going into the subsurface and it's staying there for weeks to months. That is a key distinction that is part of the explanation for why at those high elevations we can support so much more plant biomass and why we have streams flowing at least for part of the year. It's because of that water that moves into the subsurface and persists there for long periods of time. So this is one example of a type of experiment that we can do now and that we can do again in 2 years and then again in 4 years and then again in 6 years and see how the results change after this soil has gone through weathering processes, after we have introduced vegetation, and after the hill slope begins to evolve through time. All right, so I'll wrap up here just by pointing out that LEO is one part of what is really an amazing facility at Biosphere 2, where we do research and also education and outreach. So LEO is unique in that it's meant to be a tool for research for the entire scientific community, not just people at the University of Arizona. All the data that's generated from those thousands of sensors that we have goes online immediately. And scientists from around the world can access that data. They can come here and work on proposals with us to do different types of experiments. So this is really meant to be a resource for the entire scientific community. And we've already generated a lot of interest. We're collaborating with groups in the US, also from Canada, and also from Italy already. B2 is also an excellent place for education and outreach. We have about 100,000 visitors a year that come there to see what's going on. And we're also very involved with the undergraduate community at the University of Arizona. I'm guiding research projects for 2 undergraduates at the moment. This summer, we're going to have a summer science academy where we've recruited students from middle schools and high schools throughout the country to come here and spend a week with us working on a project together so that they can learn about the scientific process. So LEO is one part of the Biosphere 2, which I think is just a really phenomenal facility to advance earth science research and also education. And so if you want to learn more, we have a website where there's lots of tremendous videos that I mentioned that Shipper did that will show you how this site was constructed, that entire process, or better yet, come on up to B2 and do a visit and see it all for yourself. So I'll stop there, and thanks for your attention, and looking forward to some good questions.